Hi, I'm Alex, and in this video, I'm going to talk about the Watson and Crick model of DNA. This is Watson and Crick. In 1953, they discovered the model of DNA, also known as bead-form DNA, a double helix with some unique characteristics that I'll show you in a second. First, let's talk about the composition of DNA. DNA is composed of nucleotides, which includes a 5' carbon, a base, and a phosphate. The 5' carbon helps distinguish if a strain is RNA or DNA. DNA has deoxyribose. You can tell by the hydrogen attached to the 2' end, while RNA contains ribose that has an OH at the 2' carbon. Now that we know the composition of DNA, let's talk about base pairing. It occurs in the center of the double helix where two nucleotides pair using hydrogen bonds. There are 4 nucleotides in DNA: adenine, thymine, guanine, and cytosine. Adenine and guanine are known as purines because they have 2 rings. Thymine and cytosine are pyrimidines who only have 1 ring. But why is knowing what a pyrimidine and purine is important? A pyrimidine pairs with a purine, but only one purine goes with one pyrimidine. Adenine pairs with thymine, and cytosine pairs with guanine. These pairs are held together by hydrogen bonds, which occurs when there is a weak interaction between positive and negative charges. G and C pair with 3 bonds, and A and T pair with 2 bonds. These bonds help support the helix, but are not the main force of stabilization. Just talked about base pairing. Let's look back at the model. As you can see, the bases are stacked upon each other. This is known as base stacking. With the help of the phosphate backbone, the bases are lined up and stacked on top of each other, creating the sequence for the DNA. For example, the sequence of this segment of DNA is AGTC for one strand and TCAG for the parallel strand. Bases are hydrophobic, and base stacking maximizes hydrophobic interactions. But why do we care about that? Well, the hydrophobic interactions are the main source of stabilization of DNA. Base stacking also gives us 10 base pairs per turn. This DNA also has a phosphate backbone, which connects the base pairs and allows for base stacking to occur. The backbone is held together by phosphodiester bonds and is hydrophilic. Due to the twisting from the plectonic coil, the DNA has major and minor grooves. Major grooves are 3.4 nanometers long, and minor grooves are 0.34 nanometers long. Protein binding mainly occurs on major grooves. The direction or handedness of DNA is also important. You can tell the direction by looking at where the backbone is pointing up. Watson and Crick DNA is right-handed. The backbone is going upwards in the right direction. Now that you have seen right-handed DNA, let's look at some examples of left-handed DNA. Here are some left-handed DNA pictures I have found from recent news articles. So if we look at the direction of the backbone, it is pointing up and towards the left, and you can see this in all the pictures. You can see the same thing from this model found in an article and this t-shirt that I found on Kohl's. The backbone is going towards the left, making them left-handed. And there you have it. That is Watson and Crick's model of DNA.